starting today, we're starting at interphase. During interphase, a cell does all of the things that a cell normally does. If it's a heart cell, it's constantly pumping blood. If it's a muscle cell on an arm, it's helping to contract and relax, allowing you to move the bones so that as a whole organism you can move. If it's a nerve cell, it's sending signals and carrying electrical and chemical signals throughout your body to allow you to respond to stimuli. Okay? However, sometimes cells wear out. They get old. They need to be replaced. That's going to happen. You need to have cell division. You have to have a cell become a new cell. Sometimes you want to grow, which means you want to add new cells. Right now I'm old. I'm only pretty much replacing the cells I've got. Maybe not even all my brain cells. I'm not as with it as I used to be, but we try. All right. But for you, you're still growing, so you're still adding cells. So besides doing what a cell normally does in its life cycle, and this is the longest phase of a cell cycle because that's where its normal functioning happens. The other thing it's doing is either getting ready to replace itself or getting ready to divide to add growth, to add new cells. Just to give you an idea of how often this happens, you start as one single fertilized cell inside your mother's womb, and you are now trillions of cells big. Okay? That's how many times this process happens. That's why it's a very important part or cellular process to understand. So besides carrying out its normal function, the last thing a cell has to do in interphase is get ready for cell division. A couple of really important things happen. One, the genetic material in the center starts to condense. Okay, we start to see it. Right now we call this darkened structures, we call it chromatin. It's like a jumbled ball yarn mess of genetic material. It also copies itself, right? So you have in every cell you have a complete set of chromosomes to carry out every function and build any structure that is you. But every cell doesn't do that. It's called specialization. Cells pick and choose which parts of information they're going to use. But the point is, every cell will have an exact copy of every other cell's genetic information. So in order for that to happen, at the end of interphase, the genetic material, in this case DNA, gets copied. The other thing that happens is the centrioles have to copy themselves because every cell is going to need centrioles, and the centriole's job, it's one of the cellular structures we haven't talked a lot about because its job is really only purposeful during cell division, is to take this jumbled copied mess that you're seeing here and divide it out into two sets so that each cell is going to get a copy of this. Okay? So that's pretty much right at the end of interphase, and now we're ready to start cell division. And during cell division, a big part of that is a very special process called mitosis. And a lot of times people interchange that and they say mitosis is cell division. It's not really. It's just the major part of it. And what happens during mitosis is we're going to take this jumbled mess and get it divided out into two sets so that we're ready to put it into the two new cells. And so the first part of mitosis is called prophase. And this is the hardest to recognize or talk about because so many things happen at once. So I'll try to get there for you. So in order for this jumbled mess to get divided, the first thing I'm going to talk about is the fact that we need to have this nuclear membrane break down. Okay, so the nuclear membrane breaks down so these things can get moved. Now you might be saying to yourself, wait a second, these things do not look like that. Well, one of the things that happens between interphase and by the time you get to prophase is you can start to see the individual chromosomes. And the chromosomes, because they've been copied, are actually linked together with the identical chromosome. And so these two chromosomes together are called sister chromatids. I don't know, why do they always talk about the female as the sister, and at the end we're going to have 2 new daughter cells? I'm not really sure how that all happened, but nice nod to the females. Okay? So these are sister chromatids. These are the same things that were here. They've just condensed further, and we can see them individually. And this needs to happen in order for them to get organized. Now, the centrioles, because they're going to organize them, have to have a way to control them. And so they create these spindle fibers. And I'm going to wake you up here. Yahoo! Cattle and pull them and get them wherever they want to go. So that's kind of what these spindle fibers are for. And the centrioles also move to opposite ends because, again, we want to be able to divide this out. We've got a jumbled mess. We want to pull one set to one side and one set to the other side so that we can organize these 2 complete sets of chromosomes. So, just to recap, nuclear membrane breaks down. Centrioles form these spindle fibers, or lassos as I like to call them. They move to opposite ends. The chromatin has condensed to form chromosomes, and because we have identical copies attached, they're called sister chromatids. And the last thing is, these sister chromatids are attached at a middle point, this little yellow dot called the centromere, and that just means the center. And that's the end of prophase. The rest gets a little easier. Metaphase is another second stage of mitosis, and it is, you can see that because the sister chromatids are lined up at the middle, metaphase middle. They call this region the metaphase plate, okay? And again, we're getting ready to divide up one set one way and one set the other way. So if you understand the goal of mitosis is to get these 2 new sets of chromosomes into 2 new nuclei, you know that immediately the next phase has to be to pull them apart. And that's what happens in anaphase. The sister chromatids are pulled to opposite ends. For that to happen, the spindle fibers had to shorten. And look at what else is starting to happen. The cell is starting to pinch. We're starting to form 2 new cells. We had 1 cell, we're going to 2 cells. Cell division. Okay? So, now that we've got these full set of chromosomes divided, we're really ready to move on, and we've got things organized. Yeah, whatever you see there, grab it. Okay? So now that we've got the organization happening, we don't need the spindle fibers anymore, and we're ready to reform the nuclear membrane. We've got 1 nucleus here with a full set of chromosomes, 1 nucleus there with a full set of chromosomes, and the cell's pinching further. This is the end of mitosis. Mitosis's goal was to take the jumbled mess that was made during interphase, and mitosis starts, and once we get divided into 2 new nuclei, mitosis is ended. But look, cell division has not. So the last phase of the cell cycle is called cytokinesis. And that's where we take everything else the cell's going to need to live and make sure that gets divided up. The cytoplasm, the organelles like mitochondria, Golgi apparatus, endoplasmic reticulum, vacuoles, all of these things, divide that into the 2 new cells. The cell membrane finishes pinching off, and voila, cell division. We went from 1 cell to 2 cells, and each of them has their own set of chromosomes, and that's it. That's the end.